antigen-antibody reaction, broadly they say that any antigen-antibody reaction, the common property, the general properties are, it has to be specific. It involves the specific epitope of antigen binding to specific paratope of antibody. And there are various non-covalent bonds, interactions takes place. They sometimes ask, when an antigen binds to its antibody, which and all are the various bonds by which they are bound to each other? Answer will be various non-covalent bonds like hydrogen bond, hydrophobic bond, electrostatic bond. One more aspect what they ask is, what is the difference between affinity and avidity? See, for example, if an antigen has got multiple epitopes and when it binds with multiple antibodies, when an antigen has got multiple epitopes, when it binds with multiple antibodies, affinity is defined as the bond between single epitope with its antibody, whereas avidity is the total bond, the total strength between all the epitopes with all the corresponding antibodies. So this is the difference between affinity and avidity. Affinity is the strength between single epitope with its antibody, whereas avidity is the total bond between all the epitopes with its corresponding antibody. And one more principle of antigen-antibody reaction is they work on the principle of Marak's hypothesis, Marak's lattice hypothesis. What is lattice hypothesis? They say that when antigen reacts with antibody, the antigen reacts with this antibody, say that antigen-antibody reaction occurs best when the numbers are equal to each other. I will tell you one example. Suppose you take a test tube, take patient serum. Patient serum will have plenty of antibodies. Yes? Then you serially dilute. You serially dilute the patient serum with normal saline. You serially dilute the patient serum with normal saline. So gradually the more antibodies will be there in the first tube. When you serially dilute, the antibody level will come down. Okay. After that you add equal amount of antigen to all the tube. You add equal amount of antigen to all the tube. So which tube the numbers are equal? Middle tube. Middle tube the numbers are equal. So they say that the antigen-antibody reaction occurs at its best. Agglutination will be properly, it will occur, or precipitation will occur properly. That means the lattice structure which is formed by the binding of antigen-antibody, that will be firm when the numbers are equal to each other, which is called as zone of equivalence. zone of equivalence. Whereas the reaction fails to occur, the reaction is weak or fails to occur if the numbers are excess, either antibody excess or antigen excess. Both the conditions the reaction fails to occur. Antibody excess is before the middle tube or after the middle tube. That is why it is called as pro-zone phenomenon. Pro-zone phenomena is antibody excess before the zone, whereas antigen excess is called as post-zone phenomenon. Whereas the reaction occurs only in zone of equivalence. So there are three zones. The reaction occurs only in zone of equivalence, whereas pro-zone and post-zone, both the conditions the reaction doesn't occur properly. Okay, so this all these are about the antigen antibody general principles. Then we will discuss about types of antigen antibody reactions. The types of antigen antibody or reactions. First we will discuss about the conventional reactions, the conventionals and newer reactions. Like that we can classify into two halves. Conventional reactions are like precipitation reaction, agglutination, neutralization, complement fixation. All these are conventional antigen antibody reactions, traditional reactions. Newer reactions are like ELISA, all that, what we will discuss a bit later. ELISA, immunofluorescence test, okay, radio immunoassay, Western blot, all these are rapid test, all these are newer methods. So let us discuss the conventional methods first. To start with, conventional method to start with, precipitation reaction. Precipitation reaction is when a soluble antigen reacts with its antibody at a particular pH, temperature and electrolyte. It forms insoluble precipitate, and this insoluble precipitate will be formed in the form of flocule if you use a liquid medium. If the medium is liquid, then flocules are formed. In the form of band it will be produced if you use a gel containing medium. This is the definition of precipitation reaction. And accordingly, precipitation reactions are defined can be classified as reactions in liquid medium and reaction in gel containing medium, that is precipitation in gel. Liquid medium examples are ring test. Ring test is Lancefield grouping, and Ascol is thermoprecipitation test. Lancefield grouping is done for Streptococcus, and Ascol is thermoprecipitation test is done for anthrax. It's very old test, not done now. Anthrax. Then slide flocculation test and tube flocculation test. Slide flocculation test VDRL, RPR. Tube flocculation test, Kahn test. Both are done for syphilis. Precipitation in gel. What is the problem of liquid medium? Liquid medium will become dry soon. You cannot preserve it for a longer time, and also it is difficult to interpret the flocules. Whereas if you use a gel containing medium, it is easy to interpret, and it can also be preserved. Preservation is possible. Example of precipitation in gel, there are various examples like radial immunodiffusion, Alex gel precipitation test, Aiken test to detect E. coli toxin. E. coli toxin is detected. All these are examples of precipitation in gel. Again, the problem of precipitation in gel is gel, the movement of antigen and antibody movement is extremely slow. It will take around 2 to 3 days to complete the reaction. So, in order to reduce the time, there is a modification is done, which is precipitation in gel in presence of electric current, so that the movement will be faster. This is called as electro-immunodiffusion, and the variant of this is counter current immunoelectrophoresis, rocket electrophoresis. All these are variant of precipitation in gel in presence of electric current. Okay, so this is all about precipitation. So what is the definition of precipitation? Which antigen it uses? Yes. In contrast, agglutination uses insoluble or particulate antigen. Example, there are various examples like slide agglutination test, tube agglutination test like Vidal test, then standard agglutination test, SAT. Standard agglutination test is done for Brucella, standard agglutination test, where MAT is done, microscopic agglutination test, Leptospira. Cold agglutination test, where Mycoplasma. Then Coombs test. Coombs test, it is done for RH incompatibility. It is done for RH incompatibility, and in this particular test, they say that you are detecting RH antibodies in serum. If you are detecting RH antibodies in fetal serum, you have to do a direct Coombs test. If you are detecting in maternal serum, you do a indirect Coombs test. So this is how agglutination reaction examples in Coombs test. Okay. Then comes indirect agglutination test, indirect or passive agglutination test. Which is more sensitive, agglutination or precipitation? By definition, agglutination is always more sensitive and easy to interpret than precipitation reaction. That is why always they try to convert precipitation reaction into agglutination reaction. That can be converted by using a carrier molecule like RBC, latex, etc., and you can coat the antigens on the surface of the carrier molecule. The soluble antigen can be coated on the surface of carrier molecule to make it insoluble. Such preparation, when it binds to when it is added with patient serum containing antibody, the antigen antibody reaction occurs on the surface of carrier molecule, and which is called as passive agglutination or indirect agglutination, because you are using a carrier molecule as a base. Example of this is, there are various examples. Indirect heme agglutination. If you are using RBC, it is called as indirect heme agglutination test, or even latex agglutination test for antibody detection. Okay, all these are Passive agglutination. So passive agglutination is what is coated, what is detected. What is coated? Antigen. What is detected in patient serum? Antibody. You can do reverse also. You can coat on the carrier molecule, you can coat antibodies that can be used to detect antigen in sample. This is called as reverse passive agglutination test. And if you are using RBC, it is called as reverse passive heme agglutination test. If you are using latex, it is again latex agglutination test for antigen detection. And even co-agglutination test also is an example of reverse passive agglutination test. Co-agglutination test. In co-agglutination test, the carrier will be Staph aureus, on which IgG will be coated, which is used to detect the antigens in the specimen. Fine. So this is about agglutination test. Then certain example of complement fixation tests like Wassermann test. Conglutinin test. Complement fixation test is also used for rickettsia, mycoplasma, chlamydia, brucella, and various viral diseases. By the way, of course, please remember it is obsolete. Now, complement fixation test is only there in books. That's all. It is outdated test. Similarly, neutralization test also is almost obsolete. The various example of neutralization tests like viral neutralization test to detect antibodies. Then Schick test, which is done for diphtheria. Previously, anti-streptomycin O antibody was detected by neutralization test. Now, of course, we do lattice agglutination test, but previously it was detected by neutralization test. Naegeli reaction. Naegeli reaction also is a toxin. So, the last three are the example of toxin antitoxin neutralization test. Schick test, ASLO and Naegeli reaction, all these are example of toxin antitoxin neutralization test. Okay. So this is all about conventional method. Now we will discuss about newer method. Newer method, as I told you, ELISA, immunofluorescence assay, chemiluminescent immunoassay, okay, Western blot, rapid test like immunochromatographic test and flow through assay, and immunoelectron microscopy. So like that there are various newer methods, and please remember newer methods are more, it is increasingly used than the older methods. Older method, agglutination is still in use, precipitation almost obsolete except VDRL, and CFT and neutralization tests are also obsolete. So newer methods are widely used now, and all these newer methods, they vary from each other by the molecule used for labeling. See, the principle is very simple. All the newer methods, what principles they have followed is either antigen or antibody you coat with a detector molecule. If you want to detect antigen, you coat the antibody with a detector molecule. If you want to detect antibody in specimen, you coat antigen with a detector molecule. When antigen antibody reaction will take place, because of the detected molecule you can visualize the reaction. So very simple principles they followed, and they vary, all these methods they vary from each other, either by the molecule which is used for labeling, or how you are going to visualize the reaction. How you are going to visualize, whether it is a collet whether it is a color change or it is a fluorescence, like that, how you are going to visualize the reaction. These are the point based upon which it is different. Like for example, ELISA enzyme is used to label. Immunofluorescence assay, fluorescent dye is used. Chemiluminescent immunoassay, various chemiluminescent compounds are used, like luminol, acridinium. All these are chemiluminescent compounds. Even one thing what I have left is radioimmunoassay, RIA. Radioimmunoassay, the radioactive isotopes are used to label, and because isotopes are used here, hence because of the radioactive hazard, this method is not used much, because there is a risk of radioactive hazard. Western blot also is slightly different. It is also enzymes are used, but it is slightly different. You are detecting the antibodies against individual component of antigen. Okay. So similarly, a rapid test, immunochromatographic test, they say that colloidal gold is used to label. Flow through assay, most of the flow through assay, staphylococcal protein A is used as a conjugate. So like that, various methods, various molecules are used to label. The basic principle is almost same. Okay. So this is all about